Good evening, class. I hope all goes very well. What you're looking at right now is a diagram I made to describe the area of cancer biology I very much hope we're going to examine together. So I'll just quickly review this diagram. If you check out the wavy pink arrow here, what it shows I hope it shows the progression of tumorigenesis and metastasis. That Hanahan and Weinberg paper, The Hallmarks of Cancer, the hallmarks are captured in that paper by the authors in an attempt to begin to delineate both the steps and the markers in the development of cancer. And my representation, this wavy arrow here, is an illustration, supposed to be an illustration of that. I don't know how good it is. So imagine for a moment, we'll take an example organ, the liver, and down at the bottom here are representations of classical liver cells. And what we're looking at through this flow in pink towards these more, let's call them differently shaped cells, we're looking at that event through tumorigenesis, through metastasis, to the point of the transition of a normal cell to a cancerous cell. The Hallmarks of Cancer describes what the authors regard as the hallmarks that can be used to describe both the event of transforming towards a cancer and the characteristics a cancerous cell has. The diagram also shows what we hope will happen for all of us in the presence of such events, is the encounter of cancerous cells with what are described here as naive CD8 T cells. They're members of the immune system, the white cell system. They're described as naive at this point because they have not encountered antigens. So cancer cells in this area here, cancer cells present antigens. Antigens are unique proteins on the surface of the cell, and in this case the proteins are non-self. Hopefully they'll be recognized by the immune system as non-self. I believe we have previously mentioned the distinction between self and non-self. If we haven't, we'll cover it again. In this instance, we have naive CD8 T cells. The naive refers to this, just to reiterate, the cells are naive. They have not met antigens before. They have not met, and when I use the word antigen here, it's a non-self antigen. They have not met antigens before. They are about to meet them, presented on the surface of cancer cells. And let's hope they have receptors that can recognize those non-self antigens on the surface of cancer cells. And if the cancer is going to progress and not be detected, this is the point in the process where the failure occurs, where naive CD8 T cells do not recognize the antigens on the surface of cancer cells. So in this area, that's what my diagram is attempting to show. So coming down from the top, naive CD8 T cells, they proliferate in response to the antigens related to tumorigenesis and metastasis. The antigens are unique non-self proteins presented on the surface of cancerous cells. Normal cells do not present these proteins. The antigen presentation is part of the process of the transformation of a normal cell to a cancerous cell. What will hopefully happen in that event, the event of the encounter, is there'll be a recognition of non-self, and that recognition of non-self is a proliferative signal within the community of naive CD8 T cells to expand the pool, expand the population of effector T cells that will clear the tumorous cells. So that's the event we're looking at. Tuesday, when we discuss the paper Hallmarks of Cancer, we'll be discussing this progression, tumorigenesis and metastasis, and this progression, I'm using the liver as an example, the progression of a normal cell through to a cancerous cell. The cancerous cells present antigens, non-self antigens, and hopefully we have naive CD8 T cells, white blood cells, T cells, that present receptors that can recognize those antigens. Upon the event of recognition, there's a proliferative signal that expands the population of T cells, and the T cells are killer T cells. They kill the cancerous cells at this point. The process failing is if that recognition event, the recognition event of non-self antigens on the surface of cancer cells, fails. We'll cover this again in class. I just wanted to position you a little bit in terms of the events we're looking at, and also give that paper the hallmarks of cancer, a little bit of context in terms of how it fits into the process described in this imagery. I just used the word characteristics. You can substitute the word hallmarks of cancer with the word characteristics of cancer. Remember that we're examining the work of authors. Everyone's idiosyncratic. We all come forward with different explanations for what we're trying to interpret. So they have given their paper the title, The Hallmarks of Cancer. Have a good evening. See you in class.